Welcome everyone to our last of the year Meet the DM Drug Developers webinar events. So today we are very fortunate to have Dyne Therapeutics presenting to us and showcasing their exciting updates. So a few housekeeping instructions as always, right? Ask questions live during this event. Use the Q&A function. You can send questions anonymously, or you can type in your name. And at the end, we are trying to answer as many questions as possible. A little bit about the Myotonic Dystrophy Foundation. We, of course, envision a world with treatments and a cure for myotonic dystrophy. So the mission of the Myotonic Dystrophy Foundation is community, care, and a cure. So we support and connect the myotonic dystrophy community. We provide resources and advocate for care, and we accelerate research towards treatment and a cure. Next slide. As many of you know, we provide lots of resources and support. So on our website, you find all toolkits and publications. You find information about our support programs. The website is also the site where you see our calendar of events, where you can register and be informed about events like this one. And then on our digital academy, we have hours of recordings of events like this one, and we hope to make this one available there as well within the next week. Next one. So a few notes with regards to upcoming events. We have one more Ask the Expert series. This will cover palliative care and DM on December 12th. Again, sign up on the website, get more information there. And then I would like to alert you, of course, regarding the 2026 MDF regional conferences. Try to find a conference near you and please register. Exciting programs and exciting updates. So I also would like to thank our community education sponsors. So thank you to Avidity and Dyne Therapeutics for supporting our communication education webinar series and events like this one. So let's go into the science. So here you see our updated drug development pipeline, and that's really the most objective indicator of progress and hope. Not only do we have over 20 programs focusing on clinical development of drugs for DM, but we also have 11 of them in the clinical testing phases. And I would encourage you to check in with regards to updates. Again, this is on our website at myotonic.org pipeline. When we look at clinical trial activities, we can distinguish or we can categorize them into four broad areas. And then they follow, next. They follow essentially the central dogma of biology. Going from left to right, we have the DNA with our genes, and in a process called transcription, an intermediate molecule, the RNA, is formed. And in case of myotonic dystrophy, we have, of course, a repeat expansion that then results in misfolding of this molecule. So this molecule forms now a very hairpin-like structure that attracts and traps important cellular proteins. And when they are not available, we have broad cellular dysfunction. So the first category of drug development covers the inhibition and transcription and includes components like gene editing. The second component focuses on the inactivation of these toxic RNA structures. The next one aims to compensate for trapped proteins. And the last category focuses on, on compensating for these proteins. They are no longer available because they are bound to these effective hairpin structures. So Dyne next is active in the inactivation of these toxic RNA. So today we are very fortunate to have the Dyne team presenting. We'll hear from Julia Carpenter-Conlin. She is a patient advocacy lead. So Julia is responsible for fostering relationships with patients, caregivers, and neuromuscular organizations like ours. Julia has an impressive work history and track record of accomplishments leading patient advocacy teams in the rare disease space. And she spent 14 years in national nonprofits focusing on organizational development. You can see here she spent time working with HIV patients and also with Boston's resource-poor population. Then we'll hear from Dr. Alison Kreutzer. She is the Global Medical Affairs Lead. Alison's role includes interpreting scientific and clinical data in useful ways for physicians and the community. Her career has focused on rare neuromuscular diseases, including DM1, Duchenne muscular dystrophy, limb-girdle muscular dystrophy. And cool fact, earlier in her career, Alison taught high school biology and chemistry to 11th and 12th graders. She has a PhD in molecular and cell biology and a master's degree in teaching. And then we will also hear from Dr. Shauna Anderson. She is the DM1 clinical development lead, and she's responsible for the design of clinical trials, safety monitoring, and communications of study designs and results. She's a physician scientist with technical and expert knowledge in rare diseases, autoimmune diseases, and immunology. So she graduated from Tufts University School of Medicine with a PhD in genetics and a medical degree. And she also has an extensive experience in the pharmaceutical industry where she spent over 10 years. So one more time, as always, please ask questions, submit them through the Q&A button function, and we'll try to answer as many as possible. So without further ado, welcome Dyne Therapeutics, welcome Dyne team, and we are excited about your presentation.
Thank you so much, Andy. My name is Alison Kreutzer, and as Andy mentioned, I am the Global Medical Affairs Lead for DM1 at Dyne Therapeutics. And first and foremost, I want to thank MDF for inviting us today. It's really an honor to speak with you. We appreciate your time, and we appreciate MDF for inviting us and giving us the opportunity. Our goal today is to provide two major updates. So first, we'd like to give you some of the clinical data that we've learned from the first portion of our ACHIEVE clinical trial. And second, we're very excited to share that the second portion of our ACHIEVE clinical trial is enrolling globally, including, and for the first time, we're very excited to report that we're enrolling within the United States. And we'll talk more about that trial and how to learn more information if you're interested. And finally, and I'm hoping that I continue to pronounce it correctly, we have a brand new name for our drug called Zeleciment basivarsen. I'll be referring to it as z-basivarsen for the rest of our talk, but I wanted to let you know this is a new name, but it's the same program that we've been developing for the past few years. Next slide, please. I will not read every word on this slide, but this is a legal document called our forward-looking statements that essentially states that what I'll be talking about today are our plans for the future. And while we're incredibly hopeful and excited about the future for our Dyne DM1 program, because they are speaking into the future, there are risks that it may not occur as it's planned. Next slide, please. At Dyne, it really all starts with our mission. We are on a mission to deliver functional improvement for individuals, families, and communities. And this is really woven into the DNA at Dyne, and it inspires us daily. So what does that mean, really? So functional improvement for DM1 is best achieved by addressing the root cause of the disease and across multiple organ systems. So let's go to the next slide and talk a little bit more about what this means. What is DM1? Of course, you are the experts in what DM1 means, but just very briefly, DM1 is a rare genetic condition, and it impacts muscle, including skeletal muscle, cardiac muscle, and smooth muscle throughout the body, and the central nervous system, or the CNS, which is basically the governor for your body. It sends signals and helps your body respond to signals all throughout your body. And so by impacting muscle and non-muscle, such as the CNS, it really impacts the body from head to toe. And what causes DM1? So it's caused by a change in a gene called DMPK. And essentially, this impaired DMPK causes problems in how proteins are made. Proteins are things throughout the body that do a lot of important jobs. So if proper protein production is interfered with, then that can cause different symptoms throughout the body. And because proteins do so many different jobs, it can cause many different symptoms. So our goal at Dyne is really to develop drugs that can impact all of the organs that are impacted by disease, and can really lead to functional improvement for people living with the disease. And let's talk on the next slide about how we do that. So to impact disease in the areas that matter, we had to be very intentional with how we designed our drugs. And so how did we design Zeleciment basivarsen, or z-basivarsen? So I know that we've done many presentations before to the MDF community, and there are many that talk very deeply about our forest platform. I'll only describe it briefly on this slide, but if you'd like to know more about the biology or the science, I really would encourage you to check out MDF's website. In addition to many resources, they also have links to our previous presentations from Dyne, so definitely recommend that you check it out if you'd like to hear more. But very briefly, our force platform specifically leverages a fragment of an antibody, which we refer to as a Fab, and the Fab acts like a key to unlock the different cells in the body that need drug. So our Fab specifically is the key that fits inside a TFR1 lock. And TFR1, all you really need to know about it is that it's present on the outside of muscle cells all throughout the body, and also in the pathway that leads drugs to get into the brain, which helps control the CNS. And so we use this Fab as a key to help unlock the different cells in the body where our drug needs to go. Then we have a linker, and then we have the payload, which we can just refer to as the medicine. Next slide, please. So let's talk a bit more through this video about how our drug is really designed to address the underlying cause of DM1.
DM1 is a rare disease that impacts you and your family. It affects your body's skeletal muscle, the heart, lungs, brain, gut, eyes, and other organs. DM1 affects people differently, and the extent of impact on these organs is different for each individual. In DM1, mutated genetic material that is at the root cause of disease forms large, sticky loops in the nucleus, trapping important proteins into clumps. These trapped proteins can't correctly cut and paste or splice genetic material required to make necessary proteins. Trapping leads to incorrect splicing, called spliceopathy, which is believed to cause symptoms of DM1, such as fatigue, sleepiness, muscle weakness, myotonia, gastrointestinal symptoms, cardiac arrhythmias, facial weakness, and others. Dyne 101 is a targeted medicine in development as a potential treatment for DM1. It is composed of a fragment of an antibody, or a Fab, connected to an antisense oligomer, or ASO, which is intended to target the root cause of the disease and correct splicing. The transferrin receptor, or TFR1, is found on the surface of tissues affected by DM1. For example, skeletal muscle, heart, lungs, gut, eyes, and the brain. It shuttles iron into cells to ensure their proper function. The Fab in Dyne 101 is designed to attach to TFR1 and uses it to drive delivery of our targeted medicine. Once inside, the ASO component of Dyne 101 moves into the nucleus, where it enables the release of the trapped proteins. Dyne 101 is being studied in an ongoing Phase 1/2 clinical trial to determine whether Dyne 101 is able to restore normal splicing in all tissues relevant to DM1, and whether this can lead to improvement in the symptoms experienced by participants in this trial.
So as you know from hearing about how our drug was designed, the design and the impact of the drug and how we were developing our drug and conducting our clinical trials, it's been really critical to partner with you, with MDF and with the DM1 community. And so without your input, we would not be where we are today. And we're very excited about where we are today. So we wanted to just start first and foremost to say thank you for all of your input. And also, as we jump into our clinical trials, we want to say thank you at the beginning and then again at the end to all of the clinical trial sites and all the participants and families who have participated in our trials. Next slide, please. Excellent. So let's talk a bit about those clinical trials. So first, I just want to provide an overview of the trials that are ongoing and the trials that we have planned. And then we'll talk a little bit more about our data. So the first portion of our ACHIEVE clinical trial is called the Multiple Ascending Dose, or the MAD study. This is the study which I'll provide some data for in just a moment. And it has 56 participants, ages 18 to 49. It is fully enrolled, and it was our first clinical study, first in humans. So the first time it had been tested in human subjects. The clinical trial that is enrolling right now is the second portion of ACHIEVE, and we refer to this as our registrational expansion cohort. MAD and registrational expansion cohort are a little bit of a mouthful, so I'll try to just refer to them as the first portion or the second portion moving forward. And this second portion of ACHIEVE will enroll 60 participants ages 18 to 65. And I'll talk a bit more about the design of this study as well. And then we are very excited to also be deep into the planning phases of our phase 3 study as well, which we hope will be a larger and slightly longer study, which we hope to start in early 2026. Next slide, please. So very briefly, in addition to all of the updates that I've provided so far, another thing that we're thrilled is that we were recently given breakthrough therapy designation for z-basivarsen by the US FDA. And so what does this mean? So you've seen our clinical development plan. There's quite a bit going on here at Dyne, and we truly need the advice and the feedback from regulators as we work through this program to make sure that we're doing the things that are right so that we can eventually get where we'd like to go, which is to a marketed and approved drug. And so we're very happy about this breakthrough therapy designation because it provides more opportunities to get guidance from the FDA and also potential for expedited review times once we do submit. Next slide, please. So now I'm going to go a bit deeper into the first portion of ACHIEVE, that multiple ascending dose study or MAD study. Next slide, please. We've had the opportunity to present different webinars with MDF in the past talking about the MAD study, but I believe this is the first time that we're really presenting data from this study, particularly at the selected dose. So we're going to show you what we believe is a favorable safety profile and also favorable efficacy, or how the drug is working, in the next slides. Okay, so just very briefly, what is this first portion of the ACHIEVE study, just in case you haven't had the chance to see some of our previous webinars? So who do we enroll? We enroll adult patients living with DM1, ages 18 to 49 years. And as I mentioned, this was the first time that our drug z-basivarsen has been really tested in clinical trials with human participants. And so it sounds kind of funny to say human participants, but it's the first time that we were testing with humans who were willing to join our trial. And so, of course, the primary objective for this was to understand the safety and tolerability. We also wanted to understand a lot about the drug, including how well it was working inside the body. So you know that we intentionally designed the drug to do certain things inside cells like the muscle and the brain. So we need to know inside the body, is that happening as we expect? And if it is that leading to functional improvement through different time function tests, muscle strength, et cetera? And then also very important are different patient-reported outcomes. In other words, when we ask the patients, how do you feel your disease has been impacted throughout treatment, what do they say? And so we're trying to take all of those data together to understand as much as we can about z-basivarsen and to start planning our next trials moving forward. So let's get a bit more into the data from this study. Next slide, please. As I mentioned, the most important thing for us in this study was to understand the safety and tolerability profile. And so the data that I'm presenting here are from our last safety data cut, which was April 23rd, 2025, and has been presented previously. So we are happy to report that we have not had any serious related treatment emergent adverse events. It's a little bit of a mouthful, but essentially it's the clinical way that we say that we haven't had serious related adverse events that the study doctors have attributed to the study drug. We do also look at, in addition to serious events, we look at the common adverse events. And so in this case, we're defining common as occurring in greater than or equal to 20% of the participants. And these could be participants either on placebo, which is the non-z-basivarsen, or essentially a sham or a fake treatment that we use as a control group. So it could occur in that placebo group, or it could occur in the participants who are receiving z-basivarsen. And those most common treatment emergent adverse events that we have seen are colds, procedural pain, flu, infusion-related reactions, headaches, and diarrhea. We have had liver enzyme elevations that have been observed in a minority of participants, and thankfully this has not had an impact on liver function in those participants. And we have not seen persistent related anemia or thrombocytopenia, which is a drop in certain blood counts. And again, this was from our safety data cut in April 2025, and this reflects about 1,000 doses of study drug administered. Next slide, please. Okay, so now I'm going to walk through a number of different measures that look at how well the drug z-basivarsen is working. And because I know that it can be a little bit tiresome to look at chart after chart, I'll be talking a little bit about what the assessment is and going through the charts a little bit more quickly. But do encourage you, if you have time, to go back to the recording to spend some more time looking at the charts if you want as well. So again, we're looking here at efficacy, or how well the drug is working. And one of the first measures that we're testing now, and will also be the primary endpoint in the second portion of our ACHIEVE trial, is called hand opening time measured by video, or VHOT. So what is this? So take a look at the blue box in the left-hand side of the screen. What is video hand opening time? And video hand opening time is a measurement of myotonia and also hand function, both of which can be impaired in DM1. And so essentially this test asks participants to make a tight fist and then to relax their hand. And what we're measuring in this is the time it takes to fully relax their hand. And we are specifically measuring VHOT of the middle finger, so the time it takes to fully relax the middle finger. And that is determined by a physical therapist who is reviewing the video. And faster in this case would be the ability to release your hand faster, and that would indicate improvement. So transitioning to the right-hand side of the screen, we have the graph showing data for the first 12 months. The gray line are 14 participants who are on placebo. So again, they participated in every single assessment and infusion related in the trial, but the infusion did not include study drug. And neither they nor their physicians knew whether or not they were on z-basivarsen or on placebo. The placebo period lasted 6 months. That's why the gray line kind of stops right in the middle of the chart. And then at 6 months, they had the opportunity to move on to active drug. The blue line represents the individuals who started on z-basivarsen and continued. And we are specifically showing 6 participants who started on the dose 6.8 mg per kg that we will be moving forward with for the rest of our clinical trials. And what you see in this graph is that people who were on z-basivarsen experienced improvement or benefit, which resulted in a faster hand opening time by video. Next slide, please. It was also important for us to look at strength. So is there an improvement or a change in strength for people who are participating in the trial? So let's start on the right-hand side of the screen in the blue box again. So this test has a little bit of a mouthful. It's called QMT, or quantitative muscle testing. And so what is that measuring? So we define the QMT total score as total strength across 6 muscles. This includes the upper and the lower body, proximal and distal muscles, all of which can be impacted in DM1. And so we measure this across all of these different muscles in the body, and then we generate the total score. And we refer to it as a percent predicted because this is the percent predicted muscle strength a person would have if they were not impacted by DM1. And in this particular case, higher numbers indicate an improvement in strength. And so on the left-hand side of the screen, one of the reasons why I walked through what the gray and the blue lines were is that you'll see this throughout all of our graphs here, that the gray line reflects the placebo, which was 14 participants out to 6 months. And you see that they had a slight decline in their strength over 6 months versus the participants who were treated with z-basivarsen, who were shown in blue. These 6 participants showed an increase in strength over time, and this is out to 12 months. Next slide, please. So far we've shown you video hand opening time, which measures myotonia and hand function. And then we also showed you strength. What we'd also like to know is whether or not our drug has an impact on the different activities that people do every day. And in other words, could this help a person move about their environment in a way that is meaningful to them? And so we measure this through looking at different assessments that are called timed function tests. Or in other words, we give participants a function to do, and then we time how long it takes them. And typically, if they can perform that function faster after they've been on treatments, then we hope that that means that the drug had a positive impact on their ability to do this particular function. And so this first function test that I'll talk about is called the 5 times sit to stand test. It's a little bit of a mouthful if you say it quickly. And essentially this test asks someone to sit on a chair and to cross their arms and hold their arms across their shoulders, essentially. And then they're asked to stand and sit in a cycle 5 different times. And so this test really provides valuable insight into a number of different measurements throughout the body, including lower body strength to actually stand, also core strength to help support your balance throughout, and also core strength, which helps support respiratory muscles, as well as activities of daily living. If you can imagine standing up from a chair at the kitchen table or potentially a toilet, this really reflects a movement that people need to do every day. And so again, people were asked in this test to do this movement from sitting to standing 5 times in a row without using their arms. And faster times in this case would indicate improvement. So if you take a look at the right-hand side of the screen for this chart again, similar in that gray is the placebo and blue is the treated. Similar that placebo is still 14 participants and the treated participants are still 6 out to 12 months or 1 year. And what you see here is that the placebo participants who did not receive z-basivarsen had about the same 5 times sit to stand score over the 6 months, whereas you start to see improvement in those who are treated with z-basivarsen out to 12 months. And on the next slide, we're going to take a look at one more timed function test. And this is called the 10-meter walk-run test. So what does this measure? Why is this important? So the 10-meter walk-run test is a measure of a person's ability to ambulate or to walk or run. And so in this particular test, a physical therapist or a doctor would measure out 10 meters on the floor, a flat surface, and a person is asked to move from the starting point 10 meters as fast as they can. So 10 meters is about 33 feet or half the length of a bowling lane. And the 10-meter walk-run test really can provide some valuable insight into a person's functional mobility or how easily they can move around their everyday environment. And in this case, similar to 5 times sit to stand, faster times would indicate improvement on this particular tests. And so looking at the left-hand side of the screen, again, the chart is the same in terms of how it's structured, but you see that in the gray, the participants have about the same or maybe a slightly slower 10-meter walk-run test over 6 months. And the 6 participants who were treated with 6.8 mg per kg z-basivarsen showed some improvement over the course of 12 months on treatment. Next slide, please. So we were very pleased and encouraged by the early results from the 6 participants who had the 6.8 mg per kg dose, that dose that we're planning to move forward with. And we're encouraged by the time function test and the strength and the VHAT scores that we see, as well as the safety profile. But we also want to know from participants in the trial themselves, how do they feel their disease has been impacted? And it's very important for us to understand the patient perspective as we go through our clinical trials. So we measure patient impact or patient-reported outcomes through several different ways in our trials. And we're going to show you here the Myotonic Dystrophy Health Index, or the MD-High total score. This is a patient-reported outcome that we like because it was really co-created by patients. So patients helped develop the questionnaire. And also, so this is really helping in our minds to really reflect what patients feel is important because they helped to co-create and co-develop this questionnaire. And so what is this specifically? So there's a lot of words on the left-hand side of the screen, but I'll read through it because I think it really is important to understand this, this questionnaire. So the MD-High is DM1 specific, and it was co-created by DM1 researchers and DM1 patient community, and it's designed to measure patient-reported disease impact. It measures 17 areas of DM1 health impact through different tests. So there's a short scale questionnaire. So there's basically 17 short questionnaires. Each one is measuring a different part of DM1 health, and then they come together to form a total score. So these 17 areas are mobility, upper extremity function, the ability to perform activities, fatigue, pain, gastrointestinal issues, vision, communication, sleep, emotional issues, cognitive impairment, social satisfaction, social performance, myotonia, breathing, swallowing, and hearing. And again, these were the health domains that DM1 patients said had the biggest impact on their life, which is why they're included in this patient-reported outcome. And the MD-High total score takes into account the scores on all 17 of these sub-questionnaires, and it's measured from 0 to 100. 0 reflects no DM1 disease burden, and 100 would reflect the greatest disease burden. What I'm showing you on the chart on the right-hand side, it starts at 0, not because their score was 0, but we actually are calling 0 their baseline or when they entered the trial. So we basically set their baseline as 0, and then we look at how their scores change over time. And so the lower the score is, the closer it is to that no disease impact. So improvement on the score is actually the score going down. So in this particular case, you see that the people who are treated with placebo, who are in the gray line, they had an increase of plus 1.9 points over 6 months, which would indicate a slight worsening of disease burden. Versus in blue, the participants who were treated showed an improvement in their MD-High scores over time, both at the 6-month time point and also at the 12-month time point. And so as we are really analyzing these early data, we are looking at 6 participants, and we certainly need to look at more participants who are treated with the 6.8 mg per kg, which we're doing now. But we're really encouraged because we're seeing favorable safety profile and favorable results, both on strength and time function tests, but also importantly on reports like the patient-reported outcome of the MD-High, which is where patients themselves are telling us how the drug was impacted by how the disease was impacted by their treatment. Next slide, please. So what else are we looking at in this study? So I mentioned at the very beginning of our talk that it's important to us to measure functional improvement by the impact on all the different organs that are impacted in a particular disease. And so in DM1, that is both muscle and non-muscle, such as the central nervous system or the CNS. And so this slide just looks at a report called the Christopher report, similarly to what I mentioned before, where patients reported the things that they experienced the most and also how debilitating those symptoms are. And so we just wanted to show this to you briefly because we know that both muscle and CNS impacts like fatigue, daytime sleepiness, and more all really come together to reflect some of the most burdensome symptoms. So just to reiterate that at Dyne, we talk every day about how we really need to address all the different organs that are impacted. And we know this includes muscle very importantly and also includes non-muscle symptoms. So we are doing our best to look at all different aspects of the disease to the extent that we can. And next slide, please, to show you just briefly what we've looked at so far. So these are data, again, from 6 participants who were treated with 6.8 mg per kg z-basivarsen in our clinical studies over 12 months. And I mentioned in the previous slide that the MD-High is a long questionnaire that is made up of 17 short questionnaires. And so each of those 17 short questionnaires look at different aspects of health that are impacted by DM1. And so what Dyne has done is take 6 of these subscales that look at things that are related to the CNS and ask participants how their treatment, either placebo or drug treatment, was impacted over the course of time. And again, remember, participants don't know if they received placebo or z-basivarsen. So we looked here at cognitive impairment, sleep, fatigue, communication, emotional issues, and pain, all of which we believe are related to the CNS. And we are seeing improvement from where patients started in each of these different questionnaires, which again is encouraging to us, especially as this is our first trial really looking at the impact of z-basivarsen. Next slide, please. Okay, so a quick summary, and then I'll dive into our enrolling clinical studies. So z-basivarsen, this is our investigational agent that we are studying in the ACHIEVE study. It had a favorable safety profile in the first portion, or the multiple ascending dose portion of the study. And we have so far seen multi-system improvement in function and strength. We also have heard from patients through patient-reported questionnaires that their disease had improved if they were on treatment. And we also, as I mentioned, we want to start and end with a huge thank you to the people who have participated in this study and continue to participate in this study, as well as all of the enormous dedication from the clinical trial sites and the doctors and all the people working in the studies. We know it takes a lot of commitment to be part of a clinical study, and all the data that we've shown you so far would not be possible without all of these people. Next slide, please. So let's talk briefly about the ACHIEVE Registrational Expansion Cohort, or the second portion of the ACHIEVE study. And as I mentioned at the beginning of the talk, for the first time, we are now enrolling within the United States, which we are incredibly excited about. So what is the second portion of ACHIEVE, or that registrational expansion cohort, which I know I said at the beginning is a little bit of a mouthful, so I'll keep calling it the second portion of the trial. So the first portion was multiple ascending dose. Again, we used that to establish a proof of concept and to select the dose regimen that we wanted moving forward, that we felt like would be potentially best for patients. In the registrational expansion cohort, we started this because we were so excited about the data that we got out of the first portion of ACHIEVE. And so this second portion of ACHIEVE is going to enroll more patients and globally, as I mentioned. So not just in the US, but it's also enrolling in Europe, in the UK, and Australia, and in New Zealand. And the plan is to complete enrollment in early Q2 2026. And why is this important? This is important for us because we are working very closely with the US FDA, or our regulators in the United States, and we are hoping that we can use the data from this cohort to support a potential US accelerated approval application. And if that is feasible, we will be submitting those data for a US accelerated approval application in Q3 of 2027. Next slide, please. So this is a little bit of a busy slide. I'll walk you through it, but I also want to mention that if you're interested in clinical trials, please always do reach out to your doctor or to a study doctor who can walk you through this in much more detail and answer many more questions than we'd be able to answer in a webinar today. But the second portion of ACHIEVE, I already mentioned that what we're trying to do from this study is get the data available to us so that we can potentially apply for accelerated approval within the United States. And so this study, if you are interested in the study, there's approximately a 2-month screening period. And we have what we're calling a 3-to-1 randomization in this study. And what that means is that for every 4 participants who enter the study, 3 of them will be randomly assigned to receive z-basivarsen, and 1 of them will be assigned to receive placebo, which does not have active drug within it. And similar to the first portion of our ACHIEVE study, this will be an IV infusion every 8 weeks for all of those participants. And this will occur, this placebo-controlled period will occur for 24 weeks, after which everyone, including the participants who are on placebo, would have the opportunity to move on to active drug, or z-basivarsen, at the 6.8 mg per kg, which is that same dose that I was showing you data from in the previous slides. So we are planning to, just a little bit more, thank you. So we're planning to enroll 60 participants. We have increased the upper age limit. So previously it was 18 to 49, but for this portion of ACHIEVE, it'll be 18 to 65. Again, I mentioned the placebo control period will be about 6 months or 24 weeks, and participants have a 1 in 4 chance of receiving placebo. The next parts of the study that people would roll into will include no placebo. So anyone who starts on placebo could move into the open label extension and the long-term extension where they receive active study drug. Also, once participants move past the placebo-controlled period, those study visits do get less frequent over time as well. IV infusions are every 8 weeks. And what are we trying to measure in this study? So it's very important that we continue to monitor safety, of course. Also, our primary endpoint is going to be that video hand opening time of the middle finger that I showed you previously. We hope that will be an early responder to treatment of the underlying disease. We will also continue to measure strength, impact on strength, and also impact on time function tests, such as the ones you saw before, and patient-reported outcomes, or how patients are saying that their treatment, either placebo or z-basivarsen, impacted their disease. And we do want to mention that the study protocol does include needle biopsies, and we appreciate people who are willing to have those biopsies who participate in the study, because that really does help us to assess the impact of what's going on inside the muscle, or that piece of the tissue that we're taking in the needle biopsy, because as this is our first study, the ACHIEVE study is the first study, it really helps us understand whether or not we're having impact on that underlying disease. Next slide, please. So if you are interested in clinical trials, first and foremost, MDF has an excellent study and trial resource center, and we've had the link here. I'm sure you've probably seen it before, but just in case you haven't, we recommend that you check out all the awesome resources that MDF has available there. If you are specifically interested in ACHIEVE, we do have a QR code on the screen, which can take you to our clinicaltrials.gov page. This clinicaltrials.gov page can give you a lot more information about the second portion of Achieve that is enrolling now, including in the United States. We also just wanted to remind you that your doctor or your neurologist is always your best resource. So feel free to talk to them about your interest in Achieve, ask about enrolling sites or if they're enrolling. And you can also reach out directly to a study doctor. So if you check out that clinicaltrials.gov website, you will find contact information for the different sites that are available. We also recommend that you check out that site frequently because as new sites are added, they get added to the site. So the site does get updated as new sites are added. So as of today, within the United States, we have sites available in Texas, Iowa, and Indiana. But again, keep checking out that page because more sites could be added. And if you don't live right next to the site, but you're interested in participating, travel assistance is available. We also have an email at clinicaltrials@dynetx.com, which you can email if you have additional questions. Next slide, please. So what are our next steps for Dyne? Next slide, please. So I've mentioned the first portion of the Achieve study is fully enrolled, and we've showed you the proof of concept study data from that portion of the trial. We've also talked quite a bit about how the registrational expansion cohort, or that second portion of the Achieve trial, is now enrolling globally, including the United States. And we hope to use these data to ask the US FDA for a potential accelerated approval with the potential of submitting that application in Q3 2027. And where else are we going? So we are hoping to have a planned phase 3 study begin in early 2026. This study, we hope, will be global. It will be designed specifically to confirm multi-system functional benefit in participants. So sites in the United States will be included, but many other countries around the world as well. And again, this is slated to begin in early 2026. So I don't have as many details to speak with you today, but please stay tuned. Please stay close to MDF because we'll be sharing many details with MDF as we move forward. And we're really excited to tell you more about that study soon. And we do know that many of you have been asking as well about pediatric DM1 patients, and we are very much hoping as well in the future to have studies for DM1 pediatric patients. So again, please stay tuned to MDF, where we'll be providing more updates as our clinical development plan evolves. Next slide, please. And this is the summary for our webinar. I will pause here because I want to make sure that we have plenty of time for questions to try to answer your questions. But also, I just want to take a moment again to say thank you to people who have informed the clinical development plan and to people who have participated in the clinical trials. We are really grateful. And the phase 3 study in particular, as well as our other studies, when you see that, you will see that people in the community informed how that protocol will move forward. And we really appreciate that input. Next slide, please. Thank you again to MDF. Thank you to you. And I've just put our clinical trial email here as well, clinicaltrials@dyne-tx.com, in case you have questions or want to connect in the future. But I'll pause here and turn it back over to MDF. And thank you so much for your time and attention.
Thank you very much, Alison, for this great presentation, and thank you also for your really great and nice efforts to explain what the respective measurements in the trials are. So we have some questions here, and we have questions from when people registered. So let's just start. So you used the term registrational expansion cohort. Can you, for people not familiar with this terminology, can you explain the term registrational in a little bit more detail?
Yeah, that's an amazing question. And I should have mentioned at the beginning, I'm sorry, that my colleagues Julia and Shauna will also be joining me. So we have a lot of expertise in the room. I'm counting them in that, not me. Just kidding. And so the three of us will be chiming in to help answer questions if you hear additional voices. But it's a great question, Andy. Thank you. So registrational expansion cohort is definitely a mouthful. Basically, very briefly, what this means is that we are hoping to be able to use the data from this portion of the study to speak to the US FDA, who are the US regulators, to ask them if it's appropriate for us to have an accelerated approval approval for z-basivarsen in the US. So that's why we refer to it as registrational. And then the expansion portion is coming from the fact that we were just so excited about the first portion of Achieve, that multiple ascending dose cohort, that we're expanding it in the second portion. So that's where the registrational and the expansion come from. But essentially the goal is to speak to the FDA and to hope to use those data for a future accelerated approval should it be appropriate.
Great, excellent. Thank you very, very much. So along those lines, let's stay with the mechanics of trial activity. So there's a question. Can you explain a bit more in detail the difference between the extension phase and the phase 3?
Yes, that's an excellent question. So that long-term extension portion of Achieve. So essentially when people start either the first portion is fully enrolled, but also the second portion, there's that portion of the Achieve trial that has that placebo-controlled period. And so that's when participants could be assigned to either placebo, which is no z-basivarsen, but they're doing everything else in the trial, or they could be assigned to the z-basivarsen. And so what happens after that placebo-controlled period is that we really want everyone to have the opportunity to then have active treatment should they be eligible and should they choose. So the long-term extension period is kind of a technical way to say that people are still in the study. They're still in the Achieve study, but they're all eligible to get z-basivarsen. And we are still having study visits. So they still go to their study doctor and they still do have different assessments and things that we're measuring. So we can get some of that long-term data collection, both from safety, which is critical, and then also from how the drug is impacting them through different functional and strength tests. So they are still doing some study visits, although in that extension period, the visits do become a little bit less frequent than it was. I believe it's every 6 months in our long-term extension period. And that could differ based on who you are or based on what portion of the trial that you're in, but it does get less frequent. And then the phase 3 study will actually be a totally different study altogether. It'll be a different protocol, different clinical trial sites, and participants there will have the opportunity, it most likely will be placebo-controlled, and then there most likely will be an opportunity to roll into a period where everyone is eligible for drug, but really more details around that are still being formulated because that protocol isn't final. So we would encourage you to move, just to wait briefly until we have more information around that.
Great. And just for clarity again, because there was another question that just came in. So is the use of the term second portion equivalent to the phase 2 of the trial? So again, right, it refers to the registrational expansion cohort of the study.
Beautifully
what
said.
you just explained. So can you comment on the exclusion and inclusion criteria of this registrational expansion cohort? So what's different? Is it the same? There are questions around that.
Yeah, definitely. And I am so thankful to have our clinical development lead here, Dr. Shauna Anderson. Would you like to take that question?
Sure. Thank you very much. And we appreciate the invitation. So the inclusion-exclusion criteria for the registrational expansion cohort is similar to what was in place for Achieve. We do have some extensions. So, for example, we are allowing, it's a broader age category than what we previously had in Achieve. And we also have removed some of the other assessments and some of the other things that are required, prior to being selected for the study. However, I will say that given that it is an extension of Achieve, we could not drastically change from the initial study to the phase 2. It is, though, we are looking forward to the phase 3, where we are going to be potentially looking at a slightly different population, and those inclusion-exclusion criteria will follow along.
If it's safe to say, Dr. Anderson, that people can check out clinicaltrials.gov for some inclusion-exclusion criteria from Achieve 2, but also reaching out to a study doctor if you had specific questions about your scenario or who you are, or whether or not might be appropriate, talking to your doctor or to a study doctor. And their contact info is on ct.gov. That's probably the best way, because we know everyone's situation can be super unique around what's appropriate or not appropriate. So speaking to that physician would probably be the best way to get more info.
Yes. That is definitely going to be your best bet, to go to your own doctor or to the clinical trial doctors. And as the new sites are opening, they are being loaded onto clinicaltrials.gov.
Great. So let's kind of as a summarizing question for the for site selections. Maybe could you give the audience a very quick primer and how do you select sites where and what are criteria that fall in? Because we get a lot of questions. When is the trial coming to Canada? When is it coming to South America? When is it coming to you name it? So maybe you can give a very quick and concise primer on this one.
Of course. So the main thing that we are interested in when we select clinical sites are going to be that the site is capable of running the study in a manner in which it is there's good clinical practice, good research practice that is occurring at that site. So that means that they have to have in general, a history of having run, very good sites in a highly ethical and compliant manner. Beyond that, whatever the study is, we want to make sure that there is a population at that site of the patients that we are interested in. And so we typically run a feasibility assessment. And so there is a survey that goes out broadly to many sites around the world. And we ask them how many patients that meet the criteria that we have listed, in general for inclusion or exclusion, do they have at their site? And so, PIs have the opportunity to review a draft protocol and to review these criteria. And then colleagues of ours that we have in medical affairs and things like that and in clinical operations, we will sit down and we will review these survey results. And the survey covers not only the patient population, but also the facilities. It also covers the additional staff and professionals that are at the sites that will allow for the study to be done in an appropriate manner. So, for example, whether or not they have physical therapists who are able to conduct all of the various assessments that we're asking for, whether they have the equipment that they need. In often cases, we are quite willing and able to provide equipment if the sites have if they have the technical expertise and if they have the patient population, and then they have a history of having done trials in a compliant manner, we will step in and often offer equipment if they don't have it, so that we'll be able to access patients and give the broadest amount of opportunity to enter into our trials as we can.
Great. Excellent. Thank you. So let's shift gear a little bit and get to one of my favorite topics. And there are many questions actually around this. So when you introduced, right, your new therapy, you said it's active in muscle, it's active in the heart, and it's active in the CNS. So we have now many questions. Your primary endpoint is myotonia, right, and relaxation measured on the middle finger. But many patients experience, let's say, CNS symptoms only, or CNS symptoms and GI issues. What is your answer to those patients, right? And in terms of the potential impact of the therapy, as well as the possible participation in trials. So patients who don't suffer from myotonia.
Oh yeah, I can start if you want to. Does that work for you? Okay, awesome. So it's an excellent question. And we do know that the presentation and how people experience DM1 can be really heterogeneous. And I think Ashana was about to really eloquently say that we want to get to as many people as possible and help as many people as possible. So our clinical trials are designed to help us try to get that answer as quickly as we can and as clearly as we can, with the goal of the data from the clinical trials being able to help us get to market, hopefully, in many places around the world. So why are we using VHOT, which measures myotonia and hand function? It's a really good question, because that means that people coming into the trial have to experience some of that in order for us to measure it throughout the trial. So VHOT and the myotonia and hand function is a very interesting, different way of looking at impact of drug, because it's really related to actually just one or two specific proteins that are impacted. So I mentioned at the beginning that protein production is impacted in DM1. And so what we hope and what we believe is that we can actually see pretty quickly a result, just in a matter of months, a result or an impact of our drug, should it be working, on this particular endpoint. And some of the other different things that we're measuring, we expect could take a bit longer, which is part of the reason that why we are really building a robustly powered phase 3 study to confirm multi-system functional improvement, which will start in 2026. But what we're really hoping is to get as early and as quick of an answer as we can. Again, all with the goal of being able to bring that drug to patients as quickly as possible. So we are definitely going to be measuring things like cardiac endpoints, GI, different patient questionnaires, different things that are coming up in that phase 3 study, because what we're hoping to see is impact across multiple different organ systems. But we do recognize that clinical trials in general, we are enrolling one specific patient population to try to answer one question, but the goal is to be able to bring our treatment to a more broad population by confirming some of that multi-system functional input through the trials.
Mm-hmm. And I think one of the things that we have to do through our trials is to establish the underlying pathobiology of this disease. So if we can get an alignment that it is splicing imbalance, this spliceopathy, this is what's causing this disease imbalance, then the premise is that if we correct that, and we can show that we are able to correct that, by looking at very easy to detect endpoints, just like Dr. Kreutzer was just mentioning. So endpoints that are quick and that are easy, that we can see in a very robust manner. Then we can then premise, well, if it's working here, and we know that this is the cause of this disease across all of these different different systems, then the premise would be that it will also work for other things as well. And we are collecting other aspects of this disease as well. We are collecting data on different things, but it is true that some of these endpoints will take longer to result out than others. It's just a matter of how these endpoints work. And so in order for us to bring these drugs to the patients who need them as quickly as we can, we often have to take endpoints that are robust, that are reproducible, that have been vetted, if you will, and have been accepted by regulatory agencies. Because if the agencies are not willing to accept these endpoints, then the road is becoming very challenging for them to accept that the data that we're getting is relevant to this patient population. So we are somewhat confined by both time and by acceptability of the endpoints that we can use. But if we can establish that this is the underlying pathobiology, then we can say, well, we are affecting this pathobiology, and so therefore it should work for other patients who may not have the exact same symptoms.
And you're obviously making an inference, right, from muscular improvements to CNS improvements, because we see questions here, right? Folks are asking, especially with regards to having a highly quantitative measurement for myotonia, and then relying on observational report outs for impact on these other scales it's softer, and many attendees here they question the underlying rigor or differential rigor. So maybe you can comment one more time on this one. Sorry for being persistent.
No, so clinical trials build upon one another. We have to establish proof of clinical concept or proof of clinical efficacy that it does work, and then we can start to build upon other endpoints and other items. So while we may not in the phase 1 or into phase 2 use tools that are as rigorous perhaps or as didactic, these are things that are being incorporated as part of our phase 3 study. And we are planning to utilize tools where we can have a much more fine-tuned method by which we can identify things such as cognitive impairment and things beyond just how patients feel about that. I will say, however, that cognitive, endpoints are across the board more challenging. They often take a little bit longer time. We are definitely measuring it because we know that our drug crosses the blood-brain barrier. And so, because it does that, and because we understand what happens when it crosses the blood barrier in muscles, it leads to splicing correction there. And so while we're not going to go ahead and do brain biopsies, the premise would be that it is also leading to splicing correction in the brain. And so in the phase 3, we will be doing much more directed, assessments for cognition, for respiratory, and for other things as well that will be in a slightly more acceptable manner.
Great. I think that was actually a beautiful summary, Dr. Anderson, in terms of the fact that clinical trials, they are not standalone, right? But knowledge study build on each other, and knowledge is integrated as we learn more. So I want to end with an answer to somebody asking for access and what is being asked in the MD High survey tool. I will take that back to our organization and see whether and how we can share this and make this available. So I really want to thank you for your presentation and the attention to explain complex presentations in a very understandable way. So thank you very, very much, Dyne team, for doing that. And seeing by the amount of questions we have, I think maybe we can plan another one of these events in 2026. I think that is really important. So if today's event spoke to you all in the audience, please consider donating to ensure longevity of these events. And then next slide. And with this, one more time, a big thank you to MDF, to the Dyne team. A big thank you also to Genevieve for making this webinar go so smoothly. And one more plea to the audience. Please consider participating in the Myotonic Dystrophy Family Registry. That's really important. It's a very important instrument to enable today's and tomorrow's trial, and we need this. Dyne team, Dr. Goetze, Dr. Anderson, Julia, thank you so much on behalf of all of us, and until next time, hopefully again in 2026. Thank you.